This session is now open. Good morning. This session is now open. If I can have the first slide, please. Good morning to all. This session is now open. Welcome to this virtual courtroom convened in honor of the International Day of Immunology 2026. Today, we gather here not merely to observe science, but to put it on trial. Before us stands a most unusual defendant, the regulatory T cells, known to all of us as Tregs. Next slide, please. For decades, Tregs have been recognized as essential guardians of immune tolerance. They are the cells that prevent our immune system from turning against our own tissues. At the heart of their identity lies the transcription factor FOXP3, a molecule so central that its discovery helped define an entire field. Next slide, please. The Treg cells maintain balance, restrain excess, and preserve what we call immunological harmony. Next slide, please. Next slide, please. Indeed, just this past year, the Nobel Prize in Physiology and Medicine honored Mary Brunko, Fred Hamsdell, and Shimon Sakaguchi, the pioneers who revealed the fundamental role of these cells in peripheral immune tolerance, cementing Tregs as one of the pillars of modern immunology. Next slide. And yet, here lies the paradox. Next slide. The very same cells that protect us from autoimmunity may also protect tumors from destruction. The same mechanisms that preserve balance may, under certain conditions, enable disease. Next slide, please. So today we ask a difficult question. Are Tregs faithful guardians of immune equilibrium, or are they, under the wrong circumstances, accomplices of pathology? Next slide, please. In this trial, the prosecution will argue that Tregs are unstable, dysfunctional with age, capable of betraying their identity, and complicit in shielding tumors from immune attack. You, our jury, the defense will argue that without Tregs, none of us would survive, that they are indispensable, and that any harm attributed to them reflects not guilt, but context. You, our jury, will hear evidence drawn from real research conducted across Latin America. You will witness data, interpretation, and debate at the frontiers of immunology. Your role is not simple. This is not a case of good versus evil, but of complexity, nuance, and biological truth. So I ask you to listen carefully, question critically, and remain open to contradiction, because in this courtroom, as in science, the most important verdict is not certainty, but understanding. Let the parties present themselves. I call now our lead prosecutor, Dr. Pablo Romero, from the Universidad Nacional Autónoma de Mexico. Pablo, please.
Thank you, Your Honor. Good morning, everyone. Your Honor, members of the jury, the prosecution brings formal charges against regulatory T cells for complicity with disease. Our evidence will show that Tregs are fundamentally unstable. They abandon their suppressive identity, they convert into inflammatory aggressors, age into dysfunction, and shield tumors from the immune cells that could eliminate them. When we correct or remove the Treg cells, the patients can improve. So, therefore, we ask this jury to find the regulatory T cells guilty.
Thank you, Pablo. We now call the lead defense, Dr. Sonia León Cabrera from Universidad Nacional Autónoma de México. Dr. Sonia will enter shortly. Perhaps the assistants, Irving and Isabella, could allow Dr. Sonia to enter the courtroom.
Hello, morning. Your Honor, members of the jury, the defense categorically rejects these charges. Regulatory T cells are literally what keeps each one of you alive at this very moment. Without them, your own immune system will turn against you. Tregs prevent autoimmunity, protects us from inflammatory devastation, and when we learn to work with them rather than against them, they become a therapeutic tool. We ask for not guilty.
Thank you, Dr. Sonia. This court will now call the prosecution testimonies. The first prosecution witness is Dr. Jefferson Leite from the Federal University of Rio de Janeiro in Brazil. Jefferson, please.
Good morning, everyone. I'm going to talk about the impacts of aging on regulatory T cell stability and function. As you know, regulatory T cells are essential to maintain immune tolerance, but what happens when these cells age? Do they remain stable guardians or do they become part of the problem? So in order to understand that, we analyze regulatory T cells in young and old mouse, and we found that old mouse have increased Tregs frequency, although these Tregs are not properly to proliferate when compared to young ones, and express less levels of suppressive molecules such as CTL4. When you look at the suppressive capacity of these regulatory T cells in a model of T cell transfer colitis, we saw that old regulatory T cells here in green were not able to properly suppress intestinal inflammation when compared to young regulatory T cells. So as you can see here in this colitis model, the transfer of aged Treg cells failed to protect Reg1 mouse against the intestinal inflammation. So these animals lost more weight, had shortened colons, and also severe histological inflammation, and endoscopy, higher endoscopy scores. Tregs are crucial to maintain tissue homeostasis and tolerance against food antigens and the commensal bacteria, as you can see here. In addition, commensal bacteria gives metabolites that sustain Treg function in the gut. However, aging disrupts gut mucosa homeostasis, shifting from a balanced microbiota to dysbiosis. And this leads to reduced microbial diversity, increased gut permeability, and pro-inflammatory environment. But do these microbiota changes in aging impairs the Treg function? In order to answer this question, we perform fecal microbiota transplantation experiments after microbiota transfer, and we look at the regulatory Treg, regulatory T cells proliferation and cytokine production. And as you can see here, aged mouse have less regulatory T cells from aged mouse have less proliferation compared to young ones. However, when these regulatory T cells from the elderly were exposed to a young microbiota, they reestablished their proliferation capacity. On the other hand, when the young Tregs were exposed to the old microbiota, they proliferate less. And this reflects also in the effector compartment. As you can see here, the effector CD4 and CD8 T cells produce more interferon gamma when compared to the young ones. But when the old effector cells were exposed to young microbiota, these old effector cells now produce less interferon gamma. And then we try to understand the mechanism behind this process. And we, in order to do that, we perform single cell analysis, and we observe that old Tregs from SPF mouse express genes related to oxidative phosphorylation, reactive oxygen species, apoptosis, TNF signaling, P53 pathway. We also found that old Tregs from germ-free mouse accumulate less DNA damage and senescence markers such as gamma-2AX and P16 when compared to old Tregs from SPF mouse. In addition to DNA damage and senescence, our single-cell analysis showed that old Tregs are enriched for TNF signaling pathway genes. And we confirm this by FACS, where we see that old Tregs express high levels of TNF receptor 1. And this was associated with increased gut TNF levels, especially after fecal microbiota transplantation of old microbiota into young mouse. And to test the role of TNF receptor 1, we transferred old microbiota into Y-type and TNF receptor 1 deficient mouse, and we observed that only Y-type recipients develop effector cells that produces high levels of interferon gamma. In TNF receptor deficient mouse, Tregs maintain their proliferation and show less levels of DNA damage and senescence, which indicates that TNF receptor 1 mediates the microbiota-driven Tregs dysfunction. So in summary, we observed that during aging, there's a dysbiosis process that is associated with increased pro-inflammatory cytokine levels such as TNF alpha, which drives instability in the old regulatory T cells. And these old regulatory T cells have more oxidative phosphorylation, senescence markers and DNA damage, which impairs their proliferation and impairs their suppressive function.
Yes. Thank you, Jefferson. We now call Dr. Eva Acosta for a brief cross-examination.
Good morning, everyone. I want to ask to the prosecutor the following question. Your data show that with aging, every immune cell population lose function, but even the context as they age change, and this may influence the Treg themselves. So can we truly blame the guardian for getting old, or the real culprit, it's the aging environment that modulates Treg function?
So, so as you mentioned, aging impacts immune populations, not all the immune populations, only the Tregs. However, Tregs occupy a central role in the tumor microenvironment, and age-associated changes in their stability, suppressive function, metabolic fitness, and disproportionately influence anti-tumor immunity. Therefore, the issue is not to attribute the function solely to the Tregs, but to consider a reciprocal interaction. So the aging tumor microenvironment is characterized, for example, by chronic inflammation, senescence-associated factors, metabolic alterations, which imposes constraints on multiple immune populations, including the Tregs. So in turn, dysfunction or destabilized Tregs may further exacerbate immune imbalance, for example, and facilitate tumor progression. So therefore, Tregs should be viewed as a critical regulatory node within an aging microenvironment rather than solely driver of immune dysfunction.
Thank you, Jefferson. We now call the prosecution witness number 2, Dr. Pablo Romero from the Universidad Nacional Autónoma de Mexico. Please, Pablo.
Yes, Your Honor, thank you very much. I am sharing my screen now, and I will try to convince the jury that regulatory T cells are guilty for being unstable. So I will show you some data about this instability, and I will try to point the fact that you have to remain stable in order to regulate the immune responses. So, Your Honor, can you see the slides? Okay, let me just share my screen. Okay. Yes, so as I was saying, I will try to convince you, yes, that these regulatory T cells can be, in fact, guilty, and I will show you some data about it. Okay, so first of all, since the discovery of regulatory T cells, they have been identified as mainly unstable. Some previous work done by the group of Professor Bluestone showed that actually when you make an unstable Treg, where you can remove the expression or downregulate the expression of FOXP3. They can become these ex-FOXP3 cells with a very low expression of CD25 that have a high expression of CD127, for example. But the main fact from this discovery is that they can produce interferon gamma and they can produce interleukin 17. And they can also lose the suppressive function, mainly also showing that after this loss of the FOXP3 program, they can be found mainly in autoimmunity targeted organs like the pancreas in the case of diabetic mice. Also, these ex-FOXP3 regulatory T cells can arise from both from natural Tregs or from peripherally induced regulatory T cells. So from Professor Blueston's group, this was some of the first evidence that showed that this epigenetic instability is driven by the methylation of the TSDR region of FOXP3. So this has a very pathogenic potential. These ex-FOXP3 T cells can be very pathogenic, very inflammatory, and they can modulate or cause autoimmune disease. This plasticity and heterogeneity of regulatory T cells has many phases now discovered. They can begin as a canonical regulatory T cells that mainly express FOXP3s that express CD25 and that have a full demethylated TSDR region, providing suppressive capacity with a very important production of immunoregulatory or anti-inflammatory cytokines like IL-10 or TGF-beta. They afterwards can become a fragile regulatory T cells that, for example, can be a Th17-like T cell that has a decreased expression of FOXP3 and can start to express some inflammatory transcription factors like ROR gamma T. They can start to have partially methylated TSDR and also have a mixed profile where they can, while providing or producing a bit of anti-inflammatory cytokines, also producing inflammatory molecules like IL-17. Afterwards, they can become these ex-regulatory T cells where they have lost all the expression of FOXP3. They have a highly methylated TSDR region and producing a lot of pro-inflammatory molecules. So we have just published a review where we show this and where we show how this regulatory T cell plasticity can be mediated by a lot of cytokines from the environment, but also by other factors, like, for example, metabolic cues or other factors that can lead to this loss of stability on FOXP3. And for example, there are two diseases where we would like to show that these regulatory T cells are mainly altered. And these are psoriasis, which is inflammatory disease from the skin, where you can see a lot of keratinocyte proliferation and a lot of lymphocyte and neutrophil infiltration. The other disease where we would like to show some instability and show some plasticity on these regulatory T cells is ankylosing spondylitis. And this is a disease where the patients can have arthritis, but mainly they have new bone formation in the joints, and particularly in some places called entheses. That is where the ligaments or tendons are inserted. So in this review that we published in Frontiers in Immunology, Your Honor, you can find all the information regarding this plasticity and showing, for example, that these Th17-like regulatory T cells can be studied in a deeper detail to show how they can alter the mechanism. And for example, some data from a group in Oxford from this publication from Davide Simone studied at single cell level these regulatory T cells and showed that although we have a lot of canonical Tregs in the synovial fluid of patients with ankylosing spondylitis, you can also have a lot of heterogeneity and you can have a lot of other regulatory T cells, like for example, can express this marker called KLRB1. KLRB1, as you can see here, is one of the markers that is expressed on the cell membrane after ROR gamma T and IL-17 expression. So this CD161 can therefore identify these Th17-like regulatory T cells. And also this data showed that the CD161 positive regulatory T cells have a lower Helios expression, showing that they probably have a very decreased suppressive function. We also showed that these classical and non-classical regulatory T cells are increased in patients with ankylosing spondylitis, and also that as long as they are losing the suppressive capacity, and as long as they are expressing more CD161, they can decrease the expression on FOXP3. Therefore, I would like to say, and I would like to convince everyone, that regulatory T cells are unstable and that they can participate actively in the inflammatory environment of these diseases. Thank you, Your Honor.
Thank you, Pablo. We now call Dr. Sonia León for cross-examination.
Thank you, Dr. Romero. However, the Treg Th17 balance is disrupted in inflammation, but restoring that balance is also a therapeutic target. Does that not prove we need functional Tregs nor their elimination?
I mean, yes, we can say that we can stabilize them. I think that if you convince us that they can be stabilized, that would be a very good option. But from my point of view, I think they are very difficult to stabilize. And I think that, yes, I mean, you can, first of all, try to become these cells to be good again and before eliminating, but I don't know. I will let the jury to decide.
Okay, we will see.
Thank you, Dr. Romero. Before we move to the defense testimonies, I would like to remind the audience, that is our jury, that your opinion matters here. So we have the chat open in YouTube where you can put your questions that will be selected, one for the defense, one for the prosecution at the end of this trial, that will help us make the decision, help you make the decision. So please put your questions in the chat in YouTube. This court now calls Dr. Sonia León Cabrera from the Universidad Nacional Autónoma de Mexico to the stand. Sonia, you have 4 minutes to present your evidence in favor of Tregs.
Okay, thank you. For years, regulatory T cells have been viewed as accomplices in cancer, but that's only half the story, and in many contexts is the wrong half. Without Tregs, the immune system doesn't protect, it destroys. Tregs are mediators of immune balance, and their key identity marker is FOXP3. They migrate to the tissues to suppress excessive T helper response, maintaining immune homeostasis. They prevent excessive inflammation, they preserve tissue integrity, and they enforce immune tolerance. The importance of Tregs becomes even clearer in the intestine. This is one of the most immunological shielding environments, constantly exposed to microbiota, diet antigens, pathogens, and Tregs are essential to maintain barrier function and prevent chronic inflammation. They control excessive immune responses and protect tissue integrity. So Tregs not shut down immunity, they calibrate it. So the question is not whether we need Tregs or not. Of course, we need it. The real problem is their instability. Under inflammatory conditions, there are many signals such as IL-4 and IL-13 through STAT6, and also epigenetic regulation that make dysfunctional Tregs. An important region for control FOXP3 expression is TSDR region, whose methylation makes unstable FOXP3 expression. Our work addresses this challenge by targeting Treg stability. We show that STAT6 inhibition combined with target demethylation of the TSDR region using a CRISPR-Cas1 system improves Treg function. STAT6 inhibition stabilizes FOXP3 expression and improves the suppression capacity of Treg cells, enhance PD-1 and CTLA-4 expression, reduce DMT1, and makes epigenetic stabilization, improving control of inflammation. So functionally, this leads to a better control of inflammation and improved therapeutic potential in models of colitis and inflammatory disease. We are not eliminating Tregs. We are reprogramming them to function properly. Thank you.
Thank you, Dr. Sonia. We now call Dr. Jefferson Leite for cross-examination.
Okay, thank you, Your Honor. Sonia, I have a question for you. You are good for stabilizing Tregs in cancer, but in the tumors you study are not those same stabilized Tregs also suppressing anti-tumor CD8 T cells? Where is the line between therapy and complicity?
Yes, it's a very difficult barrier, but I think it's very important for Tregs the environment, and if we develop strategies to make, for example, iTregs more established, we can use them for therapy, and we are developing a methodology to do the expression of FOXP3 and the methylation of state to TSDR region may control, and you can use them for therapy. Of course, this is a huge work, and we are working in that because in cancer, as you know, Tregs are helping tumors, no? And we have to be very careful to design the exact moment when we can use them.
Okay, thanks.
Thank you.
Thank you, Sonia. We now call Eva Acosta Rodríguez de la Universidad de Córdoba in Argentina to present her evidence in defense of Tregs. Eva, please. Eva? Apologies, Eva, we cannot hear you. If you could please restart. Thank you.
Okay, I think now, yeah.
Thank you.
So sorry. Thank you, Your Honor, and good morning to everyone. Member of the jury, of the audience, I appear today as a witness for the defense. My name is Eva Acosta Rodríguez, and I study regulatory T cells. ¿Sigue sin escuchar? Sorry, do you hear me?
Yes, we hear you, Eva.
Okay, okay. Sorry, I start again. So thank you, members of the jury. Thank you, Your Honor, to the audience. I appear today as a witness for the defense. My name is Eva Acosta Rodríguez, and I study regulatory T cells in the context of Chagas disease. The parasite that causes, in the context of the trypanosoma cruzi infection, the parasite that causes Chagas disease. I have heard the data from the prosecutors and also the data of my colleague from the defense. And I understand why these cells can look suspicious. But let me tell you that the story is more complex and far more interesting than expected. So just to put you in context, T. cruzi infection has two very distinct chapters, so two different stages. During the acute phase, in the first week, parasites are abundant in blood. The immune system mounts a strong Th1 inflammatory responses, and there is a transient tissue damage. So the body is in a full battle mode. But eventually, then comes the chronic phase, a month to years later. And the parasite is largely controlled, but persists in tissues. Also, there is a low-grade inflammation, and over the time, there is a progressive organ damage, particularly in the heart, in the muscles, in the intestine, and also in the nerves. So this is one of the causes, for example, of Chagas cardiomyopathy. However, it is not known how the regulatory T cells are working throughout these stages. This question was largely unanswered when we started our research, our investigation. And the answer, as I will show you in the few slides, turned out to be much more complicated and amazing. So let me start with the evidence, an evidence that a prosecution could use against my client. During the acute phase of the infection, regulatory T cells are even reduced in proportion. So here we see in an experimental model how at days 12 and 20 after infection, there is a reduction in this T cell that we characterize by the expression of FOXP3. But when we check for these Tregs, using an RNA-seq to look at the characteristic, we see that there is an increased expression of many markers, including markers of activation and also Th1 polarization, including TBET and CXCR3. But don't be fooled by the numbers nor this instability. These are not unstable T cells. They are just specialized. Also, these cells upregulate CD39, that is an enzyme that degrades ATP generating adenosine, that is an immunosuppressive. So we ask what were these cells doing, and we depleted this experimentally using a genetic model in which we can use diphtheria toxin to specifically deplete of Tregs. And when we depleted Tregs, what we saw is a boost in CD8 T cell response and also a reduction in parasite burden. So I have to admit it, they look bad for the defense. Remove the T cells, the Tregs, get a better parasite control. The prosecution will say guilty as charged. But I will ask you to reserve your judgment because this is only the first half of the story. When we move fast forward toward the chronic phase and we let the infection progress up to 120 days after infection, we see a completely different story. Something remarkable has happened. While Tregs remain low in secondary lymphoid organs such as the spleen, and here you see the flow cytometry dot plot and here the quantitative data. However, what we observe is that this Treg accumulated in different tissues. In this example, in a skeletal muscle. So Here you see the quantitative data. And what is most important, we analyze this cell and we observe that this cell show a hybrid phenotype. They keep some of the markers of specialization into Th1, but also they acquire many attributes of tissue repair. For example, the upregulation of ST2 and Calergy1 markers. And this is characteristic of a population that it's able to promote tissue repair. So again, we wanted to understand the role of these cells, and we depleted regulatory T cells by injecting diphtheria toxin, in this case in the muscle, so at the local level where these cells were accumulated. We were successful by this injection by reducing the Tregs, and what we observed is that when we depleted the Tregs, we observed after 1 and 2 weeks of infection a dramatic increase in tissue damage, showing more calcification, necrosis of the fibrillar cells, also fibrosis, and also changes at ultrastructural levels, such as mitochondrial accumulation in places where they do not support the muscle function. So in the chronic phase, these same cells that look suspicious or accomplices during the acute phase now are actively repairing the tissues. And also, to our surprise, we detected that when we depleted these regulatory T cells in the muscle, there was a clear and significant increase in the parasite burden in the tissues. So these cells were not only repairing, but also keeping the parasite in control. So a completely different context, a completely different story. More importantly, when we decided to do another experiment, another piece of evidence that I think it is critical. What happened if we deplete this Treg in the acute phase and then wait into the chronic phase? Of course, we knew that if we deplete the Treg in the acute phase, we have more parasite-specific CD8 T cell and less parasite. However, when we follow this same animal into the chronic phase, we observe that then the Treg could not longer accumulate in the skeletal muscle. These mice suffer from more tissue damage, and also they have a worse parasite control into the tissues. So what this tell us? That the Treg present during the acute phase were not just suppressing immunity, they were learning, forming an imprint, establishing the precursors that you need in the chronic phase to migrate into the muscles and to become tissue protective and to control the parasite. So if you eliminate them in the acute phase, you can win that battle, but you will lose the war. So just to put you in context, I'm convinced that the Tregs are not criminal, but just learning. During the acute phase, you have young Tregs that can make mistakes. They suppress CD8 T cell responses, but then they are getting educated. And during the chronic phase, this experienced Treg arrives into skeletal muscle and other tissues, and they know exactly what they have to do. Control parasite and also repairing the tissues. Thank you, Your Honor.
Thank you, Dr. Eva. Before we proceed, we call now Dr. Pablo Romero for cross-examination. Thank you, Dr. Eva.
Yes, thank you. Thank you, Your Honor. Dr. Acosta, I have a question for you. You argue that regulatory T cells are essential in the chronic phase, but your own data shows that simply depleting them early and waiting doesn't work. Sorry, if can anyone mute, because I'm hearing an echo and I cannot ask the question. Dr. Acosta. Yes. So as I was saying, you argue that regulatory T cells are essential in the chronic phase, but your own data shows that simply depleting them early and waiting doesn't work. If young Tregs need to be educated by the acute phase to become the wise guardians that you describe, isn't that an admission that they start out as liability? What would it actually take to teach them to repair rather than suppress from the very beginning?
Thank you, Pablo, for that question. So if you allow me, I would like to present a little bit more data about that, because we have been able to address or to identify some of the signals that would allow us to educate better this Treg during the acute phase.
Yes, we're seeing your screen.
Okay, so the candidate is this this cytokine that is IL-33. IL-33, it's an alarmin, so it is a cytokine that is released after tissue damage. And as I mentioned, those regulatory T cells that accumulate during the chronic phase express ST2, that is the receptor for this cytokine. And this cytokine is also known to induce a tissue repair phenotype in Tregs. So we did it to treat infected mice very early during the infection and analyze them during the acute phase. And the first thing that we saw is that this treatment led to an accumulation of regulatory T cells with a tissue repair phenotype in many tissues, and particularly in skeletal muscle. And more importantly, this treatment and increase of regulatory T cell with tissue repair leads to an improved disease progression with a better control of tissue damage, and also to a reduced parasite burden. So we believe, we believe that this data suggests that there is a certain window in which we can somehow force or lead to this Treg to be educated in a way that they can have an experience phenotype positive for the progression of this infection.
Okay, thank you.
Thank you, Dr. Acosta and Dr. Romero. And now, before our closing argument, this court has selected 2 questions from our jury. And the first question is there any clinical evidence showing that Treg cells can be stabilized with favorable results in terms of safety and efficacy for treating diseases? I invite one of the
Hello, thanks. Yes, there are some evidence. For example, administration of low doses of IL-2 and also adoptive Treg transfer are very useful to treat inflammatory bowel disease and graft versus disease. And it is possible to use Tregs to treat inflammation. Of course, I think there are many research around the world trying to improve strategies, for example, CAR T cells, of these kind of strategies, to use them for therapy. But there are many successful evidence.
Any of the witnesses want to add anything?
Yeah, I would like to add that I think a very critical point is to understand heterogeneity in Tregs. So I think we have a lot of work to do in the different clinical context and pathology to understand the heterogeneity and which subset or population of Treg or specialization it's playing a role. And then if we really want to manipulate them, to go directly to the subset that is playing a role.
Thank you. If there are no further questions,
Yes, Your Honor. I saw that someone is asking in the public that if this plasticity and microenvironment are variables to take in account about the epigenetic of regulatory T cells. And I will say that yes, we need a lot of more studies to determine what are the factors that drive Treg instability. And mainly the studies right now are addressing how cytokines can change their phenotype, but I consider also metabolic changes, epigenetic changes, and also many other factors that we still don't know can be involved in this. Thank you, Your Honor.
Thank you very much to all the witnesses from both prosecution and defense. I think all of you presented an excellent case. But now, before this court delivers its verdict, we really want to know who our jury is. Who are you? So please open the Mentimeter link in the chat, in the YouTube chat, and we have selected 3 quick questions for you to help us understand where you are. So you can also scan this QR code that is presented in the screen right now. And please respond to the 3 questions. The first question will require a free answer because we are asking, where are you joining from today? The second question, you will select one of the options, whether you are a student, researcher, doctor, or healthcare professional, or science enthusiast, or other. And the third question is if you had heard of regulatory T cells before today. So here we have answers for the first question. It's updated in real time, as you can see. Now the second question, whether you are a student, researcher, healthcare professional, or science enthusiast. To answer this, you can simply click on the answer that appears on the answer that appears to you, or again, scan the QR code. Perhaps we have to refresh the link. Oh, there we go. So we have all groups represented here, especially researcher and scientists. Thank you for being here with us today. I have students, scientists, health professionals, and also enthusiasts, but mostly scientists. Thank you very much for taking your time to be with us today. And now the final question. So have you ever heard of regulatory T cells before today, or is this your first exposure to these important cells? Remember that you will need to use this information to give your verdict in a few seconds. Very good. So some of them know the basics. Some of them work with regulatory T cells as expected. Thank you very much. Yes, so most present people work with regulatory T cells, but many of them have heard about them, but never worked with them. And some of them heard but didn't know what they do. So I hope by now you have a good idea. So members of the jury, the time has come. You have heard the evidence. You have heard the prosecution and the defense's cases. You have seen real Latin American science presented in this courtroom today. Now you must decide the fate of regulatory T cells. The question will be presented in your screen, and you have 2 minutes to vote. Please access the Mentimeter and give your verdict of guilty, not guilty, or mixed, depending on the context. Very well. The jury has delivered its verdict. As we can see here, one person found that regulatory T cells are guilty. 7 people believe that regulatory T cells are not guilty, and the great majority, 19 people, believe on a mixed verdict because it depends on the context. As you saw today, Latin America has the talent, the patience, and the scientific question to help solve this puzzle. What you have seen today is real science done in our region on some of the most pressing challenges of immunology. Members of the jury, colleagues, and witnesses, we have heard compelling testimony from both sides. The evidence makes one point unmistakably clear. Regulatory T cells, or Tregs, are neither heroes nor villains. Their role is defined not by intent, but by context. The very same cells that protect us from destructive autoimmunity can, under different circumstances, protect a tumor from an immune attack. This duality is not a flaw, it's a fundamental feature of immune balance. The landmark contribution of Sakaguchi, Branco, and Hamsdell, recognized with the Nobel Prize in 2025, revealed that peripheral immune tolerance is not optional, but essential. Can I please have the first slide? Can I please have the first slide for the closing statement? Okay, so we'll carry on saying that our witnesses have advanced this field with rigor and creativity. Dr. Leite's research showed that aging destabilizes Tregs, yet they keep suppressing anti-tumor response. And Dr. Romero, also from the prosecution, showed that when the Treg Th17 balance breaks, chronic inflammation wins. On the defense side, we heard by Dr. León Cabrera that stabilizing Tregs controls intestinal inflammation and improves cancer outcome. And from Dr. Eva Acosta, in Chagas disease, Tregs are costly in the acute phase, but essential to protect against cardiomyopathy and inflammation. And that is where the future lies, not in eliminating Tregs, nor in blindly enhancing them, but in learning to modulate them with precision, to activate their protective functions in autoimmunity and transplantation. It's important to know their exact for these cells, to selectively restrain them within tumors and site of infection without unleashing systemic autoimmunity. This is not a case of guilt or innocence, but it is a case of understanding complexity and rising to meet it. If I have the next slide, please. So before this court is adjourned, we would like to thank very much all the speakers and also all of you of the jury who are present here today. And in a very special manner, we thank the ALACI junior community that is represented here by their president, Dr. Maria Julia Lamberti from Argentina, University of Cordoba in Argentina, for bringing this important issue to discussion today. And finally, I would like to invite you to come and join us in 2027 for the ALACI meeting in Ouro Preto in Brazil. Please put in your agenda October 18 to the 22nd in 2027. So now this court is adjourned. Thank you very much.